Hi, everybody, and welcome to this lecture. And what we're going to be looking at today is cell division by mitosis. So our learning objective is we're going to learn all of the different phases and events when it comes to mitosis. Now, where we left off last lecture, we talked about interphase and we talked about our M phases. We talked about the G1, the S, G2, and we talked about the checks. So what we're going to be doing on this lecture is we're going to be focusing on that M phase or mitosis. And we're going to see exactly all of the different steps and different portions when it comes to going through that cell division. Now, as you know, mitosis is a type of cell division right here. And basically what we're trying to do is we're trying to create one where we're trying to take one cell, in this case, the mother cell, and we're going to divide that to produce two new cells, also known as daughter cells. And these are going to be genetically identical to itself. The reason why is because mitosis is for the body. Now, we are going to cover another one known as meiosis, and that's going to be specific to sex cells. In this case, mitosis is for the body. So one way that I always use to kind of remember or differentiate between the two is mitosis sounds like my toes, which are part of your body. So mitosis, my toes deal with your body cells. So in this case, when it comes to mitosis, remember, we're taking our one cell right here, and we're going through, remember we went through our G1, our S, and then our G2. And now we're going to go through our M phase in order to go from this one cell all the way down until we have two cells, which are going to be identical to each other. Because again, whenever we're producing more body cells, we want them to be identical from the previous ones. Remember, anytime we go through this, we're going through growth or repair. So again, we want to make sure that we're producing skin cells where the skin cells need to be. We're producing eye cells where the eye cells need to be. We're producing, any type of cell where they need to be. So we're making exact identical copies of each other. So let's go ahead and take a look at the phases of mitosis. So right here, these are going to be the four phases that occur in strict sequential order, meaning that we have to go through prophase first, then we'll go through metaphase, anaphase, and then finally telophase. Now, cytokinesis is going to be the process of dividing cell contents in order to make the two cells. So again, all of these phases are going to be the portions where we're arranging, separating, and moving things. And then cytokinesis is going to be that actual physical split. Now, one way you can kind of remember the phases of mitosis is if you go ahead and take all the first letters here, we end up with P mat. So as long as you can remember PMAT when it comes to mitosis, remember mitosis is dealing with body cells. Mitosis sounds like my toes. So we're dealing with our body cells. PMAT, prophase, metaphase, anaphase, telophase for all of the different phases in order to make more body cells. And remember, the reason why I go through this is because we're taking one cell and we're trying to produce two identical. Again, cytokinesis is the actual division of that cell to make two new cells. And over here, you can see this is what all of the different phases are going to look like. You can see we have prophase over here, metaphase, where they're kind of being in the middle, anaphase, moving apart, telophase, really getting all of the contents away from each other to go ahead and divide out. And then finally, cytokinesis, where we have those two individual pieces. All right, so let's go ahead and take a look at the phases of mitosis. Now, before we can really get into all of our different phases, we need to understand and look at exactly what a cell is going to look like prior to our M phase. So looking at this over here, this is going to be the cell right before mitosis. So looking at this over here, you can see we have these centriomes, we have our nuclear membrane, and then we also have our DNA. In this case, we have our sister chromatids, which means that we have two copies of each chromosome. Because again, remember, we're kind of doubling up in order or anticipation for that cell division. So again, this is just at the end or the late phase of G2. We have it grown, it's much bigger, we have everything that we need, and we're just about to go ahead and go through that cellular division. So looking at our first phase of mitosis, it is going to be prophase. So when it comes to prophase, the cell is going to start to break down some structures and build other ones up, setting up for that cell division. Now, these are the three characteristics that come with prophase. So the chromosomes are going to start to condense and become much more visible. You can see that this right here, the chromosomes are condensing. If we go back to the previous stage, you can see that our sister chromatids are all going to be uncondensed. So they're all going to just be floating around in the nucleus. This over here, prophase, you can see that our chromosomes are condensing down. So we're going to start to be able to see them. The next thing is the nuclear membrane is going to start to break down. Now, this is important because the nucleus is all about keeping the DNA inside itself. So we need to go ahead and break that down in order to kind of separate out those DNA pieces so we can go through cell division. Because again, our end goal is going from one cell to two identical pieces. And we're going to go ahead and divide that out in order to achieve that. We're also going to have mitotic spindles, which are going to begin to form. Now, these are going to be really important because these are going to be the actual pieces that are going to pull apart the DNA of chromosomes so that each cell can have a copy. Okay, so again, these are the three major portions or steps that have to do with our prophase. So our next part is going to be metaphase. Now, in metaphase, the mitotic spindles are going to capture all the chromosomes and they're going to line them up in the middle of the cell ready to divide. So one way you can kind of remember this is metaphase. Meta, such an M, just like middle. Just like all of these are going to line up in the middle of the cell ready to be pulled apart. So when they all align in the middle here, this is going to be known as our metaphase plate. Now, this isn't necessarily a physical structure. It's just a term that we use where all the chromosomes are going to line up. Now, what's going to happen is we have our mitotic spindles. They're going to be attached. And what's going to happen is they're going to get ready to be pulled apart. Okay, so again, that's going to be really important because, again, we're trying to separate these out so we have two identical cells. In our next part, anaphase, in anaphase, the sister chromatids separate from each other and are moved towards opposite ends. So you can see how prior in our metaphase, remember meta, middle, they were meeting in the middle. You can see that we have all of our fibers pulling apart or pulling away from the center. So this is how I can kind of remember anaphase. Anaphase, A, away. They're pulling away from the middle. So that's the major characteristic that you'll notice when it comes to anaphase. Telophase. In telophase, the cell is nearly done dividing, and it's going to start to reestablish its normal structures as cytokinesis or cell division begins to take place. So remember, in prophase, we went ahead and dissolved some things. We started to build some things up in order to get ready to go through cell division, or in this case, dividing or pulling apart the cells. Now, because we were able to go through metaphase, them in the middle, we separated them out. In anaphase, remember, ana away. Telophase, we're starting to build some of those things back that we need in order to have a fully functioning cell. So in this case, our mitotic spindles, remember those things that were responsible for kind of pulling apart our chromosomes, they're going to start to break down because we don't need them anymore. Their main job was to separate that out. We've already done that, so we no longer need that. Two new nuclei are going to form, one for each sets of chromosomes. So in this case, you can see that our nuclear membrane is going to start to reform. Because again, it dissolved in prophase because, again, we needed that DNA to be free-floating so we can pull it apart. Now that we've done so, we want to recapture that and we want to make sure they're staying in one central place. So again, it's going to start to reform. And then chromosomes begin to decondense and return to their stringy form. So again, remember, in prophase, they condense down because, again, we don't want everything kind of just floating around. We want it to be nice and condensed so when we pull it apart, we don't lose anything. Now in telophase, because we've already done all that, they're going to go ahead and go back to the nucleus as it reforms, and they're going to uncondense or decondense to their original state. And then finally, in cytokinesis, what's going to happen here is it's going to actually or actively physically divide out into our two identical daughter cells. So it's going to do that by pinching in the middle here. Now, what's going to happen is this is going to be known as a cleavage furrow, and this portion is going to start to pinch apart until we have our two identical daughter cells. Okay, so just a quick summary of mitosis, because I understand that this is a lot of information kind of getting thrown at to you right away, what I've done is I've given you this chart to show you exactly the name of the different phases right here, an image to show you exactly what it looks like, and then key points as to what that phase is doing. Now, one thing I would highly suggest you do is go ahead and start this. Make sure that you have this, because this is going to be a really, really great way to kind of refresh your memory as to what all the phases of mitosis are very, very quickly, and visually see that as well. So let's go ahead and quickly go over this. So prophase. Remember, prophase, what's going to happen is the chromosomes are going to start to condense down. So remember, all that DNA in the middle or in our nucleus is going to start to condense down. So it's much easier to manage. The nuclear membrane, remember the nucleus, is going to start to dissolve. Because again, the nucleus is really, really good at keeping everything in one spot. But again, when we're trying to divide that out, because remember, we have that second set now of DNA, for our second cell, we don't want that to be kind of caged up in the middle. We want that to be free floating because we want to arrange it in certain ways so we can go ahead and divide it out. Centrosomes are going to duplicate and move to opposite poles. So remember, those are the things right here that are going to help pull apart our chromosome in order to make sure we have DNA sets for the new cells. So prophase, those are the major things we need to remember. Metaphase, remember, meta, M, middle. The major thing that we know for this one, or the major characteristic, is that in metaphase, all the chromosomes are going to line up in the middle here, known as our metaphase plate. And then again, each sister chromatid is attached to a mitotic spindle. So again, you can see that we have these here, and they're going to, again, go ahead and attach their spindles to that chromosome. So this is what it's going to kind of look like right here. And then we have our fibers, which are going to pull in these opposite directions. So we can end up splitting this into two pieces. Anaphase, remember, starts with A. Ana is going to be away. Major thing to know from this is that it's just going to pull away. Again, what's happening here is we have all the stuff we need, and you can see it's pulling it away, and all of the things that are going to become this cell are on this side, and everything that are going to become this cell is on this side. Telophase. What's going to happen here is chromosomes are going to decondense, nuclear membrane is going to reform, and our mitotic spindles break down. So think about telophase as its total or completed, almost completed. It's went ahead and pulled everything out. So what's going to happen here is you can see that this is going to be one cell here, and this is going to be the other cell. So we're going to start to reform things that we previously broke down in our prophase, which specifically is going to be our nuclear membrane. And then we're going to break things down that we made here that we no longer need. In this case, our mitotic spindles. Finally, cytokinesis. It's going to actively pinch off so we can end up with those two identical pieces. What's going to happen is that pinching point is going to be known as a cleavage furrow, and that's going to separate out the two daughter cells, again, giving us two identical cells from when we started in prophase, which was one. So we took one cell, went through prophase, metaphase, anaphase, telophase, and cytokinesis in order to end up with our two identical daughter cells. All right, and that brings us to our review questions. So what I want you to do is to go ahead and take a couple seconds to pause this video and answer the questions, and then we'll check it out in the following slide. All right, welcome back, and let's go ahead and check this out. So looking at this over here, separation of sister chromatids occur in which stage of mitosis? Remember, anaphase, away. They're going to move away into opposite directions, so they're going to go to the opposite poles. Individual chromosomes condense during which stage of mitosis? Prophase. Remember, prophase is the beginning. We're going to condense that down. We're going to dissolve the nuclear membrane so we can kind of move around that DNA so we can go ahead and separate it out. Sister chromatids align in the middle of cells during which stage of mitosis? Metaphase. Remember, meta, middle. They're going to be meeting in the middle so they can go ahead and get ready to be pulled apart. The nucleus reforms and chromosomes decondense at which stage of mitosis? Telophase. Teletotal. It is almost totally completed. In this case, we're going to reform things that we previously got rid of in prophase, and we're going to get rid of things that we previously created in prophase, because again, we will no longer need those, and we will recreate things that we do need. The cytoplasm and cellular contents are divided during what part? Cytokinesis. The cytokinesis is going to be the actual divisional portion or the actual separation. What's going to happen is we are going to create that cleavage furrow. It's going to pinch and it's going to physically separate that out. So instead of that one cell kind of pulling apart, but still being connected, this is the actual physical separation of that cell, making it go from one cell into two cells. So if a cell normally has 4 chromosomes, how many chromosomes will each of its daughter cells have after completing mitosis? Remember, it's going to be 4. The reason why is because we're creating exact identical copies from its parent cell. So in this case, if we have 1 over here and it had, let's say, 10, after mitosis, we're ending up with 10 and 10. We want identical copies. So remember, it's going to go through our G1, S, and G2 in preparation for our mitosis or that cell division. So again, we're going from 1 cell to 2 cells, and they're going to be identical or exact copies from the original. What might explain the unusual pattern seen in this cell? Looking at this over here, during metaphase, one of the chromosomes failed to bind microtubules from both spindle poles. Looking at this over here, you can see that this right here is anaphase, because remember, they're pulling away from each other. And you can see right in the middle, this one separated out great. This one did as well. This one did too. But you can see here, there was a failure in that. So you can see that this is going to eventually lead to a genetic disorder, because again, now we're going to have a mismatched portion of our chromosomes. So again, you can see that this didn't separate out, because a normal one would look like this over here. But because that failed to happen, now we have more on this side right here and less on this side over here. All right, and that is going to conclude mitosis. So again, if anybody has any questions, comments, concerns, please feel free to ask or reach out to me. Until then, I look forward to seeing everybody in the next lecture.